team just arrived here in Sardinia. It's absolutely gorgeous here. You can see we're up on the mountain, we've got the sea, and we've got this beautiful landscape with amazing geology throughout the whole entire island. So we're extremely excited to see what else the island has in store for us, both in the local marine ecosystems and in the underwater cave systems. Hey, come on. The Marine Genome Project is an organization I founded about a year ago. We are dedicated to marine conservation work first and foremost, but we focus on using genetic tools and resources to do marine conservation work. It's very unique. There's not a lot of organizations in the world who even use marine genetics for any of their work, let alone specialize and focus on it. What we also try to do is we also try to blend the world of citizen science and marine science. Divers, snorkelers, swimmers all have thousands of hours and days and years of experience in the ocean. And all that information can be extremely valuable for scientists who maybe don't go out in the ocean as often, or maybe don't have as easy access, or don't have the skills and experience required to work in some of these harder environments. And so MGP is designed to blend these two worlds to better educate the general public about more complicated topics such as marine science, genetics, marine genetics, scientific diving with the scientific world. Give them opportunities to have research collected in environments that they normally wouldn't have access to, such as underwater cave systems, deep ocean environments, local marine ecosystems. And because it's fairly unique that there's really only a couple places in the world that have cave systems like that, it provides an excellent lab for us to research how life evolves, how they can adapt to changes in harsh environments and conditions, and how we can better understand even just our ocean environment. Because they provide the perfect controlled environment for us to study how temperature and salinity and pH and nutrients and light affect life. That way, hopefully, we can get a better insight of how we can protect the rest of our water world. The purpose of this first dive was to kind of get the wiggles out, get some of the kinks worked out before we start doing our project diving. We haven't dove together as a team yet, or at all, so we want to make sure that we go on a little dive just to kind of get to know how each other work. That way, when we start doing some project diving and some surveying, we work a little bit better together because we've had some time to go on some dives together. So that's kind of the purpose of this dive. And then we'll be diving the rest of the week to collecting eDNA, biological information, survey information in the cave systems, and see what we can collect. Yeah, that is really interesting. There's not too many places like that around the world that have access like that, where the caves are directly connected to the ocean, and you get in the ocean to get in the caves.
Well, there is probably some place, but here you have a whole gulf, and it's about 24 miles with hundreds of caverns and multiple serious big cave systems, talking about freshwater systems getting into the sea. And they are technically called ankyling. So it means there is an intrusion of salt water into underground freshwater rivers. On top of that, during ice ages there was multiple changes in sea level, and the result is an even more complex situation that affected the caves and affected the sea outside.
So I'm really excited to learn more about the ecosystem and the environment here, because this will be my first time here, and excited to learn more about how we can help contribute with each other and create those goals that you see in the future. We can then hopefully learn more about these ecosystems we all enjoy.
Looking forward, lots of water time.
Oh yeah, I can't wait.
In general, I think there is a lot of cave divers nowadays able and willing to cooperate with researchers and scientists. Each of them may have a different taste or some different preferences in terms of what they would like to do, because in the end it's all volunteers. But me personally, I would like to see especially the ankyalin caves, the sea caves, becoming a little bit more studied. And this kind of studies start with documentation, which puts together video, photography, survey, sampling of water and sediment, and more analysis and more cooperation between two different group of explorers, if you want. Cave diving explorers and science explorers, because in the end we want to achieve the same goals.
So eDNA stands for environmental DNA. So all living things release cells into the environment. So bodily fluids, tissues, fish scales, hair, everything gets released into the environment. So the air, the water, the soil, and it contains DNA of whatever that organism was that it released into the environment. eDNA can be used to identify species. So we would go through and we would collect some of the environment. So in this case, in the caves, we'll collect some of the water. And then all of the species that have been living in the cave system are releasing tissues and bodily fluids into the environment, into the water. And then we can use eDNA and extraction methods to take out the DNA out of that sample to identify what species they are. And then we can track and we can model it with like photogrammetry, cave surveys, or anything else to see how far they're making into the system. This week we're working in 4 different underwater cave systems. There's 4 different sites: Calaluna, Bumarino, Grotta del Fico, and Beltorrante. So these 4 sites allow us different accessibility. They all have different geological formations. They have different amounts of fresh water systems, salt, heliclines, openings where there's air pockets, different amounts of organic material, hydrogen sulfide layers. So it gives us a vast opportunity to study these different ecosystems, environments, and conditions that are in the cave systems. They're super exciting. I don't know, anywhere else where there's such variation that allows us to study how these systems work, how they're interconnected, and hosting different biological life in marine ecosystems. We're on our way right now to Bumarino. The cave system is amazing. You pull up and the water is turquoise, blue. It's really big, massive cave system. So one team is going to be collecting eDNA, and then the other team's going to be doing some surveying. So we'll film both teams, and then me and Erwan will go in between the teams and get some footage.
And perhaps find some new species.
Yeah, you never know.
Let's see.
Deer, let's go. What's fascinating about the Bumarino cave system is that the cave is constantly changing shape and form. Despite the pitch black darkness, our lights reveal ornately decorated passages. Through the cave, there are hydrogen sulfide layers that lay just above the floor. At about 1 kilometer in, we find our predetermined sites to begin collecting the eDNA samples. Here we finally have an opportunity to carefully collect samples that might help us reveal species that have never before been seen or even described. We honestly feel like we're on a different planet, and it's incredibly exciting to combine exploration and science in such a big way.
We are doing something which is unique. We are trying to do something which has never been done before. And we need to discover how to do it, creating some proper protocol. And once these protocols are actually working, then it becomes a replicable model, expanding incredibly the amount of caves and scenarios that can be analyzed.
Fabio, our captain for this week, is going to be taking us to Cala Luna. Cala Luna is a relatively new cave system in terms of geology. The next dive we have planned for today is we're going to go to Cala Luna and we're going to place some cookies where we want to survey for our eDNA collection. Cookies are little pieces of plastic that have numbers on them and some identification. A letter like this is S for survey, and then a number on each one so we can identify where in the cave system they are. So we'll go up the saltwater side, we'll place a cookie there so we can sample the saltwater, and then we'll go up to the freshwater and we'll place another cookie there so we can collect some DNA from the freshwater side. And that's what the plan for this dive is. What's different and unique about this system is it's a little bit harder to explore due to some of the paths we have to take, and it goes narrow and wider in some sections, so it adds some logistical issues with exploration. What also is cool about the system is, unlike others, when salt water and freshwater come together, sometimes you get sections that are more brackish, where we're not quite freshwater but not quite saltwater. And that provides a unique environment for understanding, okay, what species might live in this saltwater section of the cave system and this saltwater branch, and what species might live in the freshwater branch of the cave system. And because there's no inner section of it that has this brackish water, it leaves some questions about what species might be able to travel in between the two systems of saltwater and freshwater. But it's still all really exciting to study these cave systems, especially Cala Luna.
At the level of European Commission, caves are not even considered endangered environments, not because they are not, but because there is lack of data. And as a result, they cannot be defined as endangered. So any kind of information coming out of these caves is beyond precious.
They're not protected areas, or they're not seen as protected areas because, you know, us as divers are really the only ones going into the system and see the changes, or, you know, enjoying them, and that a lot of the information doesn't necessarily get out, at least it's easily accessible. And so they don't have the data to make decisions on, yeah, let's protect it, or we should regulate and protect and preserve these areas for everybody to enjoy, no matter what we're doing. And so I think that's a really important point, is that we just need to collect more information about what's going on. So yeah. The final cave system our team will be working in is called Grotta del Fico. This special location has lain hidden for tens of thousands of years, deep within the sheer cliffs of eastern Sardinia. Grotta del Fico will certainly be one of our more technically challenging dives for our teams. We plan to dive for about 2.5 hours, covering a distance of around 1.6 kilometers. Even some of the most experienced cave divers describe this particular cave system as a scary roller coaster, featuring tight passages with dramatic depth changes and sections with varying visibility. We certainly saved one of the more difficult caves for our last scientific expedition. But if we map the caves correctly and gather biological samples as far back in as possible, the rewards are potentially huge. We might just discover a new species. This system's unique because most of the cave system, over 90% of it, is mostly freshwater, which provides a totally different environment than the other cave systems. So it provides a unique opportunity to understand the different biological life that's just in these freshwater systems, and do some of the systems like Cala Luna that has freshwater passages have some of the same species that live in Grotta del Fico. We're here back at the shop. Now it's time to do a little bit more work. So we did the diving, we collected the samples, we got out, we got all our equipment cleaned. The next step is we need to actually filter our eDNA so we can get it more stable, so we can process it and extract the DNA later. Okay guys, so now that we've gone out and we've collected the eDNA samples, it's now for the next step to filter them for storage, so then we can do later DNA extraction, okay? Bottle number 1, okay? So you can hold it just like that, that'll work perfect. So you can start to squeeze it, and then keep going, because you gotta draw all the water all the way through. So keep going, pump hard. We also gotta test the water for pH, total dissolved salts, and conductivity. So all you're gonna do is submerge that little purple diode in there into the water and then tell our researcher Dave what the pH level is. The total dissolved salts and the conductivity help us understand if we're in saltwater, brackish sections or freshwater sections, and how much nutrients is in this environment. Is it fairly low in nutrients or is it high in nutrients? This all helps us understand how these biological life that lives in these systems interact with each other and in the local environment.
8.02.
Okay.
8.02.
Yeah. Now that we've got the samples filtered and they're all in pre-preserved packages, we're ready to take them back home, back to the lab, so then we can extract the DNA off of the filters. We can purify them, get them ready for sequencing, so that way we can better understand what's living in these underwater cave systems. We've seen just such amazing stuff. We've found tube worms, we've found some cavefish, we found crabs, tunicates, we've found some anemones, we've found a nudibranch. We found tons of different life living in these cave systems that potentially have never been described before or identified.
This week was beyond special. It was the start of a cooperation between Phreatic and Marine Genome Project. It's an incredible and new approach to research on DNA. Environment DNA is probably a new wave of research to better understand and identify species in different environments. It's basically trying to create a library of genetic information, and this specific library related to caves is basically not existent nowadays.
And collecting eDNA is so new, especially in these environments, that we're breaking the ground. And not only how do we do this effectively, accurately, but allowing others to participate, because the more people that are involved, especially citizens and people who are champions for these environments, the better protected we can have these environments, the more we can understand, the more we can hopefully preserve them for the future.
To me, this is a demonstration that diving is a mean to an end. So it's not just going in the water and having fun, or in a cave and having fun, but it's a way to put our skills and our capacity to the service of science and research.